Before going into the topic details, I want to bring your attention to the UPSC coaching programs offered by ClearIAS. ClearIAS offers online and offline courses to help candidates get selected for prestigious government services like IAS, IPS, or IFS. ClearIAS, Prelims cum Mains, PCM course, provides a highly effective study plan, well-organized subject-wise videos, study materials, mock exams, and guidance to cover the entire GS syllabus of UPSC CSE Prelims, Mains, and Interview systematically. As a limited period offer, there is a 40% discount on our online courses. Enroll now.
Har Gobind Khorana has an important contribution in decoding the genetic code. So, what do you mean by genetic code? We have DNA, right? DNA is important because it has the information of our entire body or entire cell for that matter. In every cell, DNA is present and DNA has the information of how that cell should work. Okay? DNA is made of nucleotides. Nucleotides are the basic components of Nucleotides are the basic component of DNA. Nucleotides are basically 4 types: adenine, guanine, thymine, cytosine. This will be dealt in detail in the biotechnology classes actually. Okay. So for now, understand that we have 4 pairs of nucleotides: adenine, guanine, thymine, cytosine. Okay. So we know DNA is a double-stranded molecule. Okay. So in this strand, we have ATGC. Then opposite will be TACG. That is, there is a complementary reaction, complementary interaction. Adenine complements with thymine and guanine complements with cytosine. Okay, this is how they interact. Okay, that is, if in one strand of DNA, we have adenine, then means complementary in the other strand, we have thymine. They interact with each other. Okay, they will be having either double bond or triple bond interaction. This is how the DNA holds its double-stranded structure. Okay, so this is the information. See, for example, one of the DNA has ATGC. This is how the information is. Okay. So, it will be like another person, it will be like TACG. Okay, this is different. The arrangement is different. Arrangement is different for different people. This arrangement ensures that what is the information. The difference in the information is what the difference in different people occurs. Okay, just remember that there is a DNA. DNA is a double-stranded molecule. And we have nucleotides, which is the basis with which the DNA is made. And we have 2 complementary DNA with complementing nucleotides. Okay. See, this ATG, they form a codon. This 3 nucleotides form a codon. You don't need to remember the term, but understand that they form a codon. Okay. This 3 codons, when ATG, ATC, like that. Okay. These are getting arranged. And they will form a protein. This codon will code for amino acids. Amino acids are basic building block of proteins. When we arrange many amino acids, we will get a protein. Okay. One amino acid means this codon, one codon. One codon codes for one amino acid. This, like that, many amino acids get accumulated or arranged to form a protein. Okay. I know you might be confused, but I cannot explain this now because it's a huge chapter. In which you need to understand from the very cell basics. Okay? It will take nearly 2 to 3 hours for that purpose. So, I cannot explain it now. Just remember that DNA is a double helical structure. It has 2 complementary DNAs, complementary structures. And in every structure, we have a nucleotide, they will bond with the complementary structure. Okay, and the three nucleotides will be known as codon. Codon, why? Because it codes for an amino acid. Like this, many amino acids are arranged together to form a protein. Clear? At least the basics is clear? Okay. So, this genetic code, how the nucleotides are arranged. Okay? How the nucleotides are arranged in the DNA, how this forms a new proteins, how this forms new amino acids. Okay? This all are depicted in a genetic code. Okay? See, in a double helical structure, we cannot see. Okay. This is a double helical structure. Okay. Some parts you see this red color. These are genes. Genes. See, if you have a brown eyes or if you have a blue eyes, this is because you have a gene in your system, in your body, because of which you are having this eyes. Okay? So, genes are part of DNA which codes for a specific quality. Okay? But the important thing is, in the entire DNA, we do not find the genes. Only in some places, these genes are present. Other places are left as it is. Okay? So, some places of the DNA or some parts of the DNA which codes for a specific quality, for example, blue eyes, is known as genes. Other parts which are not coding for anything but are required for the normal functioning is the other parts. Okay? We have exons and introns. I'm not complicating it. Just remember that the part of DNA which codes for a specific quality is genes. DNA has both genes and non-coding part. Okay? So, see, normally if we consider, the non-coding part will be different for different people. Okay? Majorly difference is seen in the non-coding part. The genes are almost similar, except for some qualities like blue eyes. Other parts of the gene will be similar. Why? Because every individual does the digestion, every individual does the respiration, every individual does the excretion part. So, for this purpose, the basic things or the basic genes will be similar. Some exceptions are additional genes like a blue eye. Eye color is something which is there. Whether it is blue or whether it is black or whether it is brown, that is a thing. That is the difference. But eye color is for everyone. Gene for eye color is for everyone. Right? So, this is being decoded by Har Gobind Khorana. Clear? Clear? Okay. So, we have a DNA. DNA consists of coding part and non-coding part. Coding part is known as genes. Genes are almost similar for every single person because they are necessary for the vital functions of human beings. Okay? Non-coding parts are usually different. Okay? So, when we decode, that is, see, here we have arrangement for eye color. Here we have arrangement for respiratory gene. Okay, like that. When we decode this, how will it occur? It will be like, see, blank spaces here. Then here it will be eye color. Then again blank spaces. Then here will be like respiratory gene. Like that we can decode. Okay. So, as I said, there is a difference in the non-coding part among the individuals, and this is used for DNA fingerprinting, paternity test like that. Okay, in forensics also. Forensic technology, this is, you might have seen that There is a DNA which is got from the hair sample, which is got from a crime scene. And in that, DNA is analyzed. And what it is, we look for the non-coding part. It is known as VNTR. We do not need it now. But yeah, the non-coding part is being analyzed because it is majorly shows difference from person to person. And as it is not important for vital components or vital functioning of a human body. Okay. This has been decoded by Har Gobind Khorana. Are you clear with this? Any doubt? Many gaps are there, I know, but I cannot explain it because of the time crunch. It will need at least 2 hours for explaining this. Basics of cell and basics of DNA and genetic code. So, decoding of genetic code, he played a pivotal role in deciphering the genetic code, which corresponds between the sequence of nucleotides in DNA and sequence of amino acids in protein. Okay? Now you know how the proteins are formed. Now you know how the DNA looks like. Okay? Now we also know how the differences be considered from person to person. In the non-coding part, majorly it is used for analysis. It is used for forensic studies, or it is used for paternity tests like that, DNA fingerprinting like that. So this was put forward by Har Gobind Khorana. He synthesized specific nucleotides, nucleic acids with known sequences and demonstrated how they correspond to specific amino acids. Similar thing. ATG codes for one amino acid. For example, asparagine. Asparagine is an amino acid. Okay. So ATG codes for, it's not like ATG codes for asparagine. This is just an example. So ATG codes for asparagine. He demonstrated that which codons codes for one amino acid. Okay. 3 nucleotides forms a codon. As I said, these 3 nucleotides forms a codon. Codon stands for one amino acid. He depicted that which codon codes for which amino acid. All other will be the combination of this one only. Okay. Next is, he studied the synthesis of nucleic acids. Okay. He studied how these nucleic acids can be synthesized in the laboratory. That is how the DNA, how the RNA can be synthesized in the laboratory. Nucleic acids is DNA, RNA. Nucleotides and nucleosides are different. Nucleosides is the basic thing, plus in which the nitrogen gets added, it becomes the nucleotides. This is the adenine, thymine, guanine, cytosine. Nucleic acids is DNA, RNA. Clear? Nucleotides and nucleic acids are different. Nucleotides is the basic part of nucleic acid. That is basic part of DNA. Adenine, guanine, thymine, cytosine are known as the nucleotides. Nucleic acids is DNA or RNA. Okay? So, what Har Gobind Khorana did is, he synthesized the synthetic or artificial RNA and DNA in the laboratory with precise sequences, enabling him to investigate the genetic code and role of various nucleotides and protein synthesis. Similar thing. The basic which I explained. He analyzed which all nucleotides will be result in the formation, which 3 nucleotides combination will result in formation of a particular amino acid. This he conducted research or study in the lab. Clear? Any doubt? Okay. Next. See, if he deciphered genetic code, or if he decoded genetic code, if he identified the amino acids formation, amino acid theory, then obviously he has important contribution in genetic engineering, right? So, his research and the groundwork for advancement in DNA sequencing, gene synthesis, manipulation of gene for various applications in biotechnology and medicine. Genetic engineering means, see, engineering, what do you do in engineering? You create new things, or you manipulate new things. You delete which is not good for things, right? Similar thing happens in the gene itself. Okay. After deciphering, what we can, I said that decoding of the genetic DNA has been done by the person. So, After this, we can identify which gene has problem or which part of the DNA has problem. Okay, that part of the DNA can be cut. Either it can be removed or can be replaced with some other part of the other gene, or it can be left as such for a normal repair. Okay, so this all are the basics of genetic engineering. Okay, I have explained this in very simple terms, but this is something which you need to study in detail in the biotechnology. Various processes are there. Okay, genetic engineering is one of the important concept which has been evolving now. With this only, in biotechnology especially, with this only various vaccines are newly being promoted, various CAR T cell therapy, right? The therapy for cancer has been initiated. Okay, so it has a wide variety of applications. Genetically modified organisms even. Okay, so contributions to genetic engineering, as I said, the genes are thoroughly studied or decoded, and after that, if there is any problem in the gene, it is being removed. Or if you want a new factor to be introduced into the DNA, a new factor was added. So manipulation of the gene is done. That is what genetic engineering is. Okay, so its contribution in genetic engineering. Next is, for this purpose, he has been awarded with Nobel Prize in 1968 for Nobel Prize in Physiology or Medicine. Okay, that is for deciphering the genetic code and interpretation of how it controls protein synthesis. For the first time, an extensive study has been made, especially in lab, by creating synthetic DNA or RNA and deciphering or decoding the entire sequence of DNA. So that is why, see, the sequencing of DNA is very important, because if you have any genetic diseases, this can be identified earlier. If you want to manipulate any function in an organism, say if you want to increase the productivity of your crop, you can add this gene to the crop. Crop means plant itself is a living organism. So definitely it is having a DNA. Okay, so you can add this gene to the DNA. For example, BT cotton. BT cotton is the only commercially accepted genetically modified crop in the country. Okay, so here what happened is, the cotton was added with a specific gene from BT. BT is a Bacillus thuringiensis. It's a bacillus bacteria. Specific gene was being added into the cotton. For what purpose? To protect it from bollworms. Bollworms are the insects which used to be feed on the cotton balls. You have the white cotton balls, right? So this has been fed by these bollworms. So it was severely affecting the productivity. So that is the reason why the BT cotton was introduced. The as such gene of the cotton was manipulated and the BT gene was added. Okay, BT gene provided the formation of toxoid. Toxoid means toxins. What does do mean by toxins? Poisonous. Okay, so toxoid means it is the subform or inactive form of a toxin. Okay, this is present in the BT cotton. When bollworm come and eat this cotton ball, this toxin enters into the bollworm, and in the bollworm's gut, this toxoid gets activated to toxins, resulting in the death of a bollworm. Okay, this is something which was added. So this was formed after this was created after the introduction or deciphering of the genetic code. That is why he has received the Nobel Prize. So this is just a simple example of how genetic modification has helped. Okay, so that is why the importance of Har Gobind Khorana. So what are his contributions? Genetic code was decoded, then contributions in genetic engineering, then amino acid theory was put forward by him for his exceptional work in genetic engineering, decoding, and also producing the synthetic DNA and conducting the various experiments. He was awarded with Nobel Prize in Physiology or Medicine.